Okay, good morning, everyone. I will start with today's session, right? Okay, so today's session is going to be our second lecture, which is called tutorial for this course. Okay, so today's tutorial is going to be mainly about sequencing, different sequencing technologies, and the output of sequencing and how it will be processed for analysis, right? I'm quite sure all of y'all are familiar with some of the sequencing technologies because you just had a bioinformatics course last semester, but I will just go through very briefly. Right, so the week before last, when I had a class, I mentioned about what is genomics, what is comparative genomics, and the different uh, methods that can be used, right? So like I mentioned, you don't really need to sequence genomes to compare them, but of course it helps. Now that sequencing has become really affordable, everyone does sequencing to do a comparative genome analysis. So before sequencing technology was available, what people did was comparative genome analysis using experimental approach, right? Using mostly DNA hybridization techniques, right? Because at that point, the technology was not there and it was also not always very cheap when it was initially introduced. And there was not abundant sequencing data available. So they were using experimental approach, right? So these are some of the methods that they were using, okay, before sequencing was introduced. So they are direct and indirect DNA hybridization techniques. So there were three: DNA hybridization, comparative genomic hybridization, and array comparative genomic hybridization. So this is the first, just DNA hybridization. So all you need is your DNA, right? Your healthy DNA or normal DNA, and you have your tumor DNA. So you need genomes to be compared, all right? So normally they use it for clinical approach. So you have normal or healthy individuals genomic DNA, and you have uh, and a person with some disease, usually cancer. So they use this genomic DNA to do this hybridization technique. So first thing they do is you label the probe with label probes for all the tumor DNA, right? And you use a different probe to tag the normal DNA, right? Like I've shown here. So the ones with tumor, they have tagged it with green fluorescence, and the ones with normal, they have tagged it with red fluorescence. And then they hybridize it to normal metaphase chromosome for about 48 to 72 hours. So they allow the DNA to hybridize to each other, right? And then they basically map the intensity of fluorescence. So if you have equal binding of the regular on probes for tumor and equal binding of probes for normal, then you will have a yellow colored intensity, which means equal binding. If you have if you have green, like really intense green, then you know that region is has only tumor probes, all right, binding to that chromosome, that region of the chromosome. And if you have red um, fluorescence, then you know that in that region of the chromosome, more normal probes had bound, right? So with this, you can actually identify mutations or regions of chromosome which has uh, tumor DNA, right? So this is one technique. Moving on, they also used to use array comparative genomic hybridization. So this is very similar to your cDNA microarray technique, where you have thousands of probes which are um, already tabbed on your sort of like your array, right? They are already immobilized on it on the surface. and then you add your genomic DNA, which is fluorescently labeled, and it is allowed to hybridize. If it's able to hybridize, right, and there will be, of course, fluorescence, and they use software to actually screen through and analyze the fluorescence intensity, right? So based on the intensity of the fluorescence, they can deduce whether there are tumor DNA or genes related to tumor or genes related to healthy individuals, right? So this is another technique. So and they always use this to identify copy number variation and also segmental duplication in chromosomes or in genes. And the last technique, which was also being used before sequencing was introduced, was CGH, or comparative genomic hybridization. It is also very similar to the array method, except that it is done on metaphase chromosomes, right? On whole metaphase chromosomes, as shown here. So you have your sample DNA and you have your reference DNA. You allow it to hybridize to metaphase chromosomes and you look at the intensity. This is very similar to your DNA hybridization technique that I mentioned earlier. The only downside of this approach is that it only permits detection of very large chromosomal or copy number changes with a resolution of 10 MB using conventional methods. And when they improved it and they came up with high resolution CGH, the resolution was still not as good as array-based techniques, and it was about 3 to 4 MB changes was was detectable, right? So these are the three techniques that researchers used to use before sequencing was introduced. Now that sequencing has been introduced, we'll move on to the next phase, right? I've mentioned here pre-NGS. NGS is next generation sequencing, right? So before NGS was introduced, Sanger was being used. And I believe you know this person, right? This is Fred Sanger. So he was the one who introduced this technology, Sanger. And he has won two Nobel Prizes. His second Nobel Prize was for Sanger sequencing technology, right? This is known as the first generation sequencing. And this was the key method that they had used to actually complete the Human Genome Project when they finished it in year 2000. At that point, they did not have NGS, so they relied completely on Sanger sequencing. And as you know, it's a PCR-based method. And I guess you're already familiar with what Sanger is, but I've just given some information here. So it relies on dideoxy nucleotides, which helps with identifying the bases in a particular fragment, right? So this is how it's done. You have your single-stranded DNA. It's amplified using dideoxy nucleotides, which are already fluorescently labeled. And once a DDNTP is added into the fragment, the polymerization is not able to it doesn't continue, right? And then you basically have to run a gel, and you will be able to read the sequence by looking at the gel electrophoresis image, right? You'll be able to deduce the sequence of nucleotides. Right. So after that, of course, this happened when we had the advent of NGS, or next generation sequencing, and we have so many technologies that have come up after that, right? And one really widely used technology is Illumina by Roche. So this is a picture of one of the sequencing machines that they have, HiSeq 2000, but of course they have a lot more sequencing machines now, and there's one new one that they have introduced, NovaSeq 6000, which has a higher capacity compared to this. Okay, and this basically started giving researchers like high throughput sequencing, tons of data, and of course, what we need now is more bioinformaticians to be able to make sense of these data. Right? This is just to show you how much data is available. So once NGS was introduced, it basically rained sequence data. So this shows how much data has been generated over the past few years. So you can see up to year 2000. Okay, the one in red is WGS, which is whole genome sequence. From the year 2000, right? Can you hear me? It shows here I've been signed out.
Yes.
All right, okay. Okay, all right. So the red line in red is actually referring to WGS or whole genome sequencing data. So you can see from the year 2000, it has increased exponentially. By year 2020, we already have more than 100,000 million sequences available in the database, right? So modern high throughput sequencing completely has changed the landscape, right? And complete, mainly accurate sequence data, much cheaper now, has now enabled precise genomic and exomic comparisons, and of course, novel analysis, insights, and visualizations. So by exomic means sequencing of protein coding genes only, all right? And if you're doing genomic sequencing, means you sequence the whole thing, including introns and all those untranslated regions, right? Right, so this is sort of how sequencing technology had evolved. We started off with the first generation, which is Sanger dideoxy sequencing, and then you have your second generation, which we are going to go through now, which is known as NGS, or next generation sequencing. We have a few technologies. Illumina is the most widely used. We have ion semiconductor and pyro sequencing. Pyro sequencing is sort of phased out. I don't think people do it any longer. If you still see articles which are published on pyro sequencing, it most likely was old data. Probably they sequenced it quite some time ago, and they're only working on it or publishing it now. And we have third generation sequencing technology, which refers to mainly Oxford Nanopore and PacBio's smart technology, right? So we'll go through NGS and third generation briefly. Right, so NGS has a lot of advantages, as you already know. Using Sanger sequencing, we will only have very short fragments, which is about 600 bases to maybe 1,500 bases fragments, which we will need to sequence tons of fragments to be able to get a complete genome, right? By using next generation sequencing, you can do this very easily as we do massively parallel sequencing in the flow cells, right? One of the major advantage of NGS is that construction of sequencing library involves just clonal amplification. In Illumina, it's known as bridge amplification, and you do not require any in vivo cloning, no transformation, no colony picking. So there's no, there's not much experiments or lab work involved. It's rather, I would say, a lot less tedious to work with NGS compared to Sanger sequencing. And it's also an array-based sequencing, so you can have massively parallel sequencing happening the same time. So your sequencing can finish in say 36 hours to 48 hours. You can finish sequencing. While if you're working with Sanger, it takes longer, as you generate short fragments one at a time, right? And with this, you will have short fragments, but thousands or hundreds of thousands at a time. Okay, right. So these are again some of the advantages. You have higher sequencing capacity with NGS. It is also cost effective, because if you're going to be working with Sanger for whole genome sequencing, you will need to spend a lot of money, as you have to clone it first. You have to pick your colonies, and your colonies will just have random fragments. You don't know if what you've picked has the same fragment as as another colony that you've picked. So it's just random, and it's just by luck if you get different parts of the genome. So of course that is one of the advantage. It's very cost effective, and it's cloning free. You don't have to do any cloning, and it has short reads. So the shorter the reads, the more accurate the data is. All right. So as I said, with Sanger, you can get up to a few hundreds to about 1,500 bases. It's still as accurate, and you don't get anything higher than that. If you get fragments which are longer than that, or bases, nucleotide reads which are longer than that, it is not as accurate anymore. Right. So moving on, these are some of the steps, the major steps involved in NGS. First is to, of course, prepare your sample. You do your genomic DNA extraction. You can use spin column technology, or you can also, of course, use phenylchloroform extraction methods. And then you have to prepare your library. And that is also a very short procedure, probably a day or less than a day, maybe just a few hours. You can finish preparing your library. Next step is to do your clonal amplification or bridge amplification, depending on the technology you're choosing. So if you are, using ion torrent, or if you're using pyrosequencing, you will have, of course, emulsion PCR for your amplification process. And if you're using Illumina, you use bridge amplification technique, right? Third is, of course, the sequencing itself. And finally is data analysis, right? So these are the commonly used, not commonly, these are the technologies which are available for NGS: Pyro, ion semiconductor or ion torrent, and Illumina. Pyrosequencing is almost phased out. You don't usually see much data coming from this technology anymore. Ion semiconductor also is not really famous anymore. Illumina is still the most widely used NGS. Right? So just to give you a brief overview, I think you have already gone through this in your last semester. So this is just to recap, right? So pyrosequencing is a technique based on pyrosequencing chemistry, which utilizes chemiluminescent stacks and enzymatic reactions. And some of the key enzymes used are luciferase and sulfur relase. And the library fragments are sequenced by synthesis. Okay, and these are the methods. So the first step is, of course, after you've extracted your DNA, is generating single-stranded template DNA library. Usually, it's just heat-induced. Then you have emulsion-based clonal amplification of the library, and then your data generation via pyrosequencing, and finally data analysis. Right. Moving on to ion semiconductor. So this technology actually combines computer software with integrated circuits, as well as your CMOS or complementary metal oxide semiconductors. You may be aware of this term, CMOS. They are actually available in your digital cameras, your laptops, your computers, and even your mobile phones. So this technology also adopts an electrochemical detection system. So each time a nucleotide is added, an ion is released, a proton is released, and the sensor actually detects the change in pH, right? So each time a nucleotide is added, it detects the change in pH, and there is a signal, right? So all of these electronics are focused on detecting and analyzing the release of a hydrogen. And I'll show you this here, right? So again, for ion torrent, you also require emulsion PCR for your amplification, your clonal amplification process. So once your emulsion PCR is done and your sequencing starts, right? When a nucleotide is incorporated, a single proton is released, and the semiconductor transistor detects this change in pH, and it actually gives out a signal. So this sequencer is basically like the world's smallest solid state pH meter, which we'll call the base, going directly from chemical information to digital information, right? So, and if you have, let's say, two exactly the same nucleotides being in the, right beside each other, I mean, like for instance, shown here, let's say you have two T's, okay, after another, and you have these identical bases. What happens is the voltage will double, right? Because it detects release of two protons, the voltage doubles, and it gets detected. Okay, and it's actually a direct detection where there's no scanning involved, no cameras at all, and no light is also involved. Okay, I've also given a YouTube link here which you can go and watch later, which explains how this works. Moving on, next is your Illumina sequencing technology. So I will go through this a bit more in detail, as this is the most widely used technology right now. Okay, so your Illumina workflow includes, the first step is your library preparation. Okay, so this is prepared by random fragmentation of your genomic DNA you've prepared, or if you're working with RNA, you have to first convert it to cDNA and then do a random fragmentation. This is followed by a 5' and 3' adapter ligation. These are your adapters. You ligate them to your fragments, and then they're PCR amplified and gel purified. Okay, so finally you have purified double-stranded DNA. They are denatured into single-stranded DNA by heating, and then you add them to your flow cell. Right, so your flow cell already has complementary probes with complementary sequence, adapters with complementary sequences, which will allow binding of your single-stranded DNA fragments to the flow cell. Okay, so they're captured by the oligos over here. And the next step is cluster generation. So each fragment is then amplified into distinct clonal clusters through bridge amplification. As you can see, this is how it looks. It actually folds over because it has a complementary adapter sequence as on the other end of the flow cell. So it folds over and your polymerase will actually catalyze polymerization, right? So this is known as bridge amplification, and you have double-stranded DNA fragments, which is again denatured, right? And the original strand is washed away, leaving only the strands that have been synthesized to the oligos attached to the flow cell. So multiple cycles of new strand synthesis and denaturation then takes place to make multiple copies of the same sequence, forming a cluster. So you have like millions of dense clusters of the same sequence being formed in order to eventually, when you sequence, you have sufficient data to make sense, right? So this is what happens. And finally, you have your sequencing. Third is your sequencing, right? To initiate the first sequencing cycle, nucleotides which are labeled with fluorescent tags and reversible terminators, which actually block the addition of the next base, primers, as well as polymerase is added to the flow cell. And when the first base is incorporated, the fluorescence is emitted. So this is exactly how it looks, right? Your single-strand DNA, your adapter sequence, and these are your nucleotides which are already fluorescent tagged with fluorescence, right? So when your first base is added, right, fluorescence is emitted and it's captured, right? So when it's captured, you get an image is acquired. So this goes on, right? And your next base is added and another image is captured, and this goes on until you have your entire fragment sequenced, right? So the fluorescence signal actually indicates which nucleotide has been added. So your every base A, T, C, G is actually tagged with a unique fluorescence. For A, it's different. For T, for C, and G, they're all different. So you know, each time you acquire an image, you know whether an A has been added or T or C or G. Okay. So once it has been added, terminator is cleaved. Only when the terminator is cleaved, the next base can come and bind, right? And this cycle continues and you get the image, which is then interpreted by a software, and a base calling is done. So when you get base calling done, you will finally get the sequence, right? So initially you just have images which are transformed into sequence data. Okay, so this is what you call base calling, and it actually gives you the sequence data at the end of it. Right, so some of the advantages and disadvantages of Illumina as well as other NGS technology is that it is very accurate because the fragments are short, reads are short, so it's very accurate, and it's high throughput. So each fragment is actually sequenced maybe 10 times, 100 times, depending on how many fragments are available in the first place, right? So usually you have hundreds of the same fragment being sequenced, right? The major disadvantage is difficulty in analyzing repetitive regions and large structural variants, right? So especially when you're working with plant genomes are known to have very long, um, long sequences of repetitive regions, long stretches of repetitive regions, which is hard to deduce whether it is just a repetitive region or multiple fragments has been sequenced, right? And if there is also structural variance, large structural variance, it's also difficult to determine as we have very short fragments. We basically rely on overlapping regions to generate a complete genome sequence. So when you have large structural variance, it's rather difficult to see whether it's placed somewhere else or actually it's overlapping. And you also, because the reads are very short, we also end up getting very fragmented genomes. Unless you do very deep sequencing, that means a lot more sequencing needs to be done to be able to get a complete genome, right? Right. So based on that, researchers actually realized that there is a need for sequencing technology which is able to sequence longer fragments, right? With Illumina, the read lengths are only about 150. The maximum is 300 bases, right? So 300, imagine nucleotide sequences which are just 300 bases, and you have bacterial genomes which are 5 million bases, okay? So you need a lot of data to be able to ensure that we get overlapping regions, 300 bases of many fragments of 300 bases which are overlapping. So with that, they realized that we really require sequencing technology that is able to sequence a longer fragment. So they came up with two new technologies. One is Oxford nanopore technology. So this was actually done by two scientists from Oxford University, right? And the second is PacBio Smart technology. And how are they different from NGS? As I said, they are, of course, they are able to sequence longer fragments, and they focus on fragments between 10,000 bases to up to 2 million bases. So there were some researchers who were able to get up to 2 million bases in one fragment, right? So this is an image of one of Oxford Nanopore's product. This is known as MinION, right? And this is your PacBio Smart technology. Okay, so moving on, this is how a nanopore or MinION sequencer looks like. It's actually as small as your pen drives. It's very small, okay, and this is how it looks, right? So this sequencing technology is based on signature electrical currents generated as a single strand of DNA passes through a nanopore, right? So this pore, right, is actually a protein pore. The pore is actually made of proteins, and its size is in nanometers. It's really tiny, and they are actually embedded in membranes. And this membrane ensures that it blocks out all electrical current on both sides, so that when you apply electrical current, the current is only through the nanopore, the pore itself, and as the nucleotide passes through, it actually disturbs the electrical signal. And each nucleotide has a distinct disturbance of electrical signal. So when an A passes, there is a slight change. When a T passes, the change is different, right? So these changes in electrical current as each nucleotide passes through the pore generates a unique signature, right? For each base, it's unique. And then we have a software which actually deduces, analyzes this and deduces whether an A passed or a T or a C or G just passed, right? And the advantage of the nanopore sequencing technology is that you obtain real-time sequencing results. So as your sequencing is happening, you can actually watch in your laptop the data being generated, right? I've also given a link here to, you can go and watch after the class to see how it works. And this is exactly how it looks, right? So this is your nanopore and this is your membrane. So this blue one is basically a protein pore where your double-stranded DNA gets unwound by an enzyme, your helicase. It basically unwinds and ensures that only a single strand of DNA goes through. And as it goes through, as I mentioned, there is some electrical current which is passed through this pore alone, right? And as it passes through, it actually disturbs the electrical signal, and it has a unique signal for each nucleotide. As you can see here, where every time an adenine passes through, you can see the electrical signal is within this range, within this light blue range, right? And every time a cytosine passes through, you can see the electrical signal actually is disturbed, and the range is somewhere between this light pink, right? And when a thymine passes through, it is around this region, the range of electrical signal disturbance, and G is around here. So with that, you can actually determine the sequence of the fragment that you have just passed through, right? So there is no fluorescence tagging involved at all. There is no camera. There's no image acquisition as well with nanopore sequencing. It completely relies on a disturbance of your electrical signal. Okay, right. The last one that I wanted to mention about is another third generation sequencing technology known as SMART, right? So it is single molecule real-time sequencing, right? From its name, you know that it also allows real-time sequencing, just like your nanopore, but it uses a different principle. Okay, it is also known as a sequencing by synthesis, just like how your Illumina sequencing technology and your Pyro sequencing technology is. It incorporates nucleotides, which are again bound to different fluorescent tags. So each nucleotide is bound to a different fluorescent tag, and you have a smart flow cell, right? And inside each smart flow cell, there are thousands of tiny pores, which are known as zero mode waveguides, right? So this is how small a smart flow cell is, the actual size. It's smaller than your finger, right? When you zoom in, this is how it looks. And it also looks like an array. So this array is what we call a zero mode waveguide. Okay, so each one of it is one zero mode waveguide. So it actually represents like one reaction chamber, and at the bottom of the chamber we have a DNA polymerase, which ensures, which helps with the polymerization as the nucleotide passes through, as your DNA fragment, your single-stranded DNA fragment passes through. So the pore size, again, this is also very tiny. It's about 70 nanometers in diameter and 100 nanometers in depth. So it is too small for light to pass through easily. So it actually creates an illuminated observation volume that is small enough to observe only a single nucleotide being incorporated, right? In addition, the fluorescent tags are also cleaved off after the incorporation of the nucleotide, so that the next one, before the next one is able to be added, right? So each time a different nucleotide gets incorporated, a different color or different fluorescence is actually detected. And for this, you require, of course, a camera to be able to capture this image, right? Therefore, the smart technology and the machine work like a giant microscope, okay? And they can you can literally see the DNA synthesis in real time as well, right? In addition, you have thousands of reactions in each zero mode waveguide that can be detected simultaneously. So you have parallelly a lot of sequencing going on in each zero mode waveguide, right? The only small catch is that the template DNA needs to be prepped or prepared into a dumbbell-shaped structure entity so that the smart bell, using a smart bell technology. So basically they use a hairpin loop fragment to ensure that it becomes closed, right? So the structure is closed and you have a circular fragment which is continuously sequenced, right? I've also given a link here. You can go and watch this link after your class to have a better idea about this technology. Right, moving on. So these are some of the advantages and disadvantages of nanopore and PacBio Smart. So basically third generation sequencing. So the major advantage, of course, is you get very long reads, allowing more overlaps with other reads and providing more accurate genome assemblies. So when you have, 100,000 bases in each fragment, you can imagine the overlapping is also uh, more accurate, right? And you'll be able to get a complete genome without, gaps, right? So imagine when you have a bacterial genome, which is a circular genome, right? A 5 million base genome. When you use Illumina, you might end up getting fragments which with gaps, which is difficult to to fix if you're going to be relying completely on Illumina sequencing, right? But with this, with third generation sequencing, you can get really long reads, which will help you with covering up the gaps in your circle, right? However, the major disadvantage is, of course, lower accuracy. As I said, the shorter your reads are, the more accurate your data is, right? And when you have 100,000 bases in each fragment, you are bound to have errors, and so you have lower accuracy, therefore lower quality score, right? This is the term they use to refer to low accuracy, which is quality score, or also known as Phred score, right? So move on to another comparison table of these two third generation sequencing technology. You have PacBio, which has SMART, right? They have two models, and this is the average read length, 10 to 15 kb. Of course, people have done longer read lengths as well, but this is the average you get. And the error rate is about 10 to 15%, right? And you get quite high amounts of data, like big outputs up to 1 GB or 5 to 10 GB of data. And this is the instrument price. This was I think 1 or 2 years ago. This is the instrument price. It probably has dropped a bit, right? And this is Oxford Nanopore, and you have also two models. I think you have more now, right? MinION is the one that looks like a pen drive, the smaller one, right? And the average read length really varies. There are researchers who have got up to 2 million bases in one fragment, but again, the error rate is high, between 5 to 15%. And with Illumina, the error rates are 1 to 3%. It's very, very low, right? And the output with MinION is a bit lower, 5 GB, but with ProMethION, you get up to 10 GB of data. It is a high throughput sequencing machine. Okay, and instrument price, this is still the same. Oxford Nanopore is still $1,000. You get one flow cell for $1,000. Okay, and so what researchers usually do is to ensure that we get a good balance of accuracy as well as getting a non-fragmented complete genome. Most researchers combine both these technologies, so they do maybe a 50 times coverage of Illumina sequencing and another 30 times coverage of third generation sequencing. So you get a good balance of accuracy and you're able to cover your entire genome. So each time you do an assembly, you will require both Illumina data as well as your nanopore data or PacBio data. And there are a lot of softwares which are able to actually help you correct all those wrong reads or errors which are found in your PacBio or nanopore data using Illumina's data, right? So this is how most researchers work when they are trying to assemble genomes or sequence genomes. They combine they basically combine Illumina sequencing and a nanopore sequencing output, right? Moving on, this is just to show you again a comparison. For second generation, you have massively parallel sequencing. However, the reads are very short, about 300 bases. That's the longest length, and it's very accurate, less than 1% error rate. 3% is, if you have really, your machine is not well calibrated or something like that, then you have issues, maybe a higher error rate, but or else you actually get less than 1% error rate. With third generation, it's actually also known as a single molecule sequencing, right? And you have really long reads, and it's very rapid. Compared to Illumina, you can do this in a few hours. You can, leave it and overnight. Let's say you prepare your library, you leave in the evening around 5 or 6. You can come back the next day morning and you have 1 or 2 bacterial genomes sequenced, okay? And with third generation, you can actually study base modification such as methylation as well for epigenetic studies. Right, so this is just to give you an overview of the different sequencing technologies which are available now and how they work and the advantages and disadvantages and how most researchers actually combine both these technologies to ensure that we get accurate genomes as well as complete genomes, right? Moving on, so once you've done sequencing, the next step, of course, is to analyze. Just having tons of data is not going to help if you do not know what to do next or how to make sense of it, okay? And so after you've done NGS or third generation sequencing, your data will be in a format called FASTQ, right? I think you are quite familiar with this term, FASTA, right? And this is how a FASTA format data looks like. Your first line, right, is it starts off with a more than sign, followed by a unique identifier or the name, right? And the second line is your sequence itself, right? So if you have gone to NCBI and you're trying to search for a gene sequence or a genome sequence, you will normally see this FASTA format data with a more than sign, and you have the name followed by the sequence, right? But when you do NGS, all right, next generation sequencing, either Illumina or if you do a nanopore sequencing or PacBio sequencing, your data will not be in this format. Instead, you will have a format called FASTQ, all right? And it looks a bit different, but essentially it gives you more information as well, okay? So this is how it looks. The first line, okay, it has 4 lines, right? 1, 2, 3, 4. The first line actually is a sequence identifier. And instead of starting with a more than sign, it starts with an alias, right? And this is followed by the name or a unique identifier, which the sequencing machine itself gives. We don't have to do that. This is how the data comes, right? Second line is your sequence itself, okay, the nucleotide sequence for the fragment. And your third line is just a separator, right, to help you view it, to give you a better viewing option, right? It's a separator. It consists of only a plus symbol. And this is followed by the final line, okay, is actually, if you see, it's just symbols, different symbols, right? And this actually coincides with the quality score for each base, right? And these symbols are actually ASCII codes or ASCII characters. And there is a table that can help you decode them. Of course, you don't need to do this manually. Once you get your FASTQ reads, there are softwares which will help you decode this and give you an idea about how good the quality of the data is, right? So these ASCII characters can be decoded, as shown here. Okay, so this is an ASCII table. So you can imagine the first few, right? This is the same sequence that I've shown earlier. The first symbol is less than symbol. So if you see less than, right, this is the less than symbol, and this is the quality score. So quality score is also known as Phred score or just Q, right? Q. So this is the score for the first base, which is T. The quality is 27. The next symbol is the more than sign. As you can see, this is the more than sign. The quality Q, right? The quality is 29. And you can keep reading, right? So each base, so with FASTQ format reads, right? It actually gives you the quality score for each base, right? So now, moving on to what does this quality score mean? You have numbers from 0 to 42 here, but each of it, what does it actually mean? Right? When you have very low quality score, it means the quality is poor, okay, and the accuracy is also lower. So a Phred quality score of 10, right, means there's a probability of one incorrect base in 10, right? So 90% accuracy. And if your quality score is 20, you are 99% sure that base is correct. Your quality score is 30, is 99.9% accuracy, 40, 99.99, 50, 99.999%. So as you can see, the higher the quality score, the higher the accuracy rate, right? So going back to this, when you actually decode what each ASCII character means, you actually know how accurate it is at each base position, right? For instance, one of it is to the fourth position or fourth nucleotide and the fifth nucleotide. The ASCII, the quality score is only 2, which means it's less than 90% accuracy at these two spots, C and A, right? So this is how you sort of decode the data that you get from your sequencing output, right? And as I mentioned, you don't have to do this manually. It is going to be crazy if you're going to be sitting and doing this manually, because each sequencing output, we get about 20 million reads, right? 20 million reads like this, right? This is just one read. And this is a short fragment. Usually we get up to 300, 150 to 300 bases. This is maybe 20, 30 base, 30 or 40 bases, right? Imagine having 20 million reads that look like this, right? So it's not possible for a human to sit and manually do this, decode and then identify which are accurate and how accurate. So we have a lot of softwares that can help us do this. So it helps us actually give an overview of the quality of the data that you just got, right? And let me see if I'm able to open this. I want to show you how it looks. Are you able to see the website that I've just opened?
No.
No, just hold on. I stop share first and I'll try to share my right. Right. So this is the software that I just mentioned in my slide. Okay, it's by Babraham Institute, and it's really a very nice tool to use. It gives you a very nice graphical illustration of how good your data is. Okay, right, this is the page. I've also given you the link there. Later after your class, you can have a look. Okay. They have actually example reports here. Okay, for good Illumina data, how it looks, for bad Illumina data, how it looks. See if I can click on good. Okay, so this is how, once you've got your sequencing data, the first step to do is to do a quality check, right? So we don't normally open and see what I showed you with the slide, I showed you how FASTQ looks. We don't normally do that at all because it doesn't, you won't understand what it means. It wouldn't make sense to you at all, right? So the first step, the moment you get your data, is to do a quality check, and we do it with a software called FastQC. There are other softwares, but this is the most widely used software, right? FastQC. And if you have a good data set, that means your sequencing data was good quality, this is how it should look, right? The first, this one gives you a summary. The first one is your basic statistics, right? It shows you what your file name is. Their file name was this, okay? What file type, this is all standard. It'll just give you conventional base calls. Encoding, it will tell you what, this is also default, Illumina 1.5. And it will tell you next total sequences. So here in their data, right, they had 250,000 sequences, okay? And here they have mentioned how many sequences are flagged as poor quality. So it's 0, which means the quality, I mean, it's quite good quality. And sequence length, that means basically shows how long your sequence is. In each fragment is only 40 bases. Hold on. Right, NCBI cannot be used to check the quality of sequences, no. It just gives you the data. You cannot know this quality, but you can always download the quality, the data from NCBI, but it needs to be in FASTQ format, and then you can put it into this software to know the quality. That can be done, right? So the sequence length is 40 bases, and it also gives you a percentage of GC. So normally when we work with genomes, we are quite concerned, or we are very interested in to know the percentage of GC. This is one of the descriptions we have in genome papers, percentage of GC. So it's also mentioned here. Of course, if you're working with RNA or transcriptome, this percentage is not at all necessary. It doesn't give you any information, but if you're working with genome, this gives you sort of an idea how GC rich your genome is. Okay, so this is the first basic stats. Moving on is your per base sequence quality. If you remembered earlier, I showed you the FASTQ format data where it had the fourth line with ASCII characters, right? And we used the table to decode what each ASCII character means. So this software does it for you, okay? It gives you an output of the per base sequence quality, right? As you saw in the summary earlier, it only has 40 bases. So this is each position, right? Position 1, position 2, position 3 in your read or your fragment which has been sequenced, up to 40 bases, right? Or 40 positions. And that is your x-axis, the position in read, and your y-axis is actually your quality score, your Q or your FRAC score. So it starts from 0 up to 38 here. So as I showed, when you have very low quality score means the percentage of accuracy is super low, right? And when you have very high quality scores up to 40, you are 99.99% sure that it's accurate, right? So this actually shows exactly that. All the reads, okay, all the reads in each position, all the way to the 40th position for the whole 250,000 sequences that were sequenced are above quality score of 28. So it's mostly about 32 and above. So the quality of the reads are really, really high. You can be 99.9% sure that the reads are accurate, right? So this is what it shows. So if you have, if your error bars go down up to the red zone, which is below 20, then you will need to do some pre-processing to get rid of the poor quality reads. Okay, since everything is above, within the green zone, then you don't have to do any pre-processing, or getting rid of poor quality reads or anything of that sort, because it's quite good quality. If you have your error bars going down to the yellow region, right, you may need to do some pre-processing, right? So this is how you read. After you've done your sequencing, you do a QC and you have an idea of your quality, right? Moving on, I'll just show you some other important things, right? Right. I've moved on to the fourth one, per sequence quality score. Okay. So with this, it also gives you the same kind of information. This is to show you that the number of sequences that was sequenced. If you remember, we had 250,000 sequences in total, right? And this is a Phred score, or the quality. So majority of the reads actually got a quality score of between 36 to 38, right? So very high quality score. This is to give you an idea of your GC content. It was already there in your summary stats in your first table, but this also shows you the same. Okay, it's about 40 something percent GC, right? Sequence length distribution will just tell you the average length of each sequence. And this was also already mentioned in your statistics table earlier, which was 40 bases. And next is the most important one that you need to look is overrepresented sequences, right? So over here, this shows no overrepresented sequences, which is good, right? That means no adapters were sequenced, no primers, Illumina primers were sequenced as well, right? This is where you need to see if there were, if there are some adapters or primer sequences which are also in your data, you will need to trim it off, all right? Because if you're going to be analyzing with the primer sequence or your adapter sequence, then your analysis is not actually accurate because it's not part of your genome. It's actually just part of your adapters or primers which are being used for your sequencing itself. Okay, so now I'll just show you how a bad sequencing data looks like. Right, so I'll just show you how a bad Illumina data looks like. Okay, so this, your per base sequence quality. Can you see how bad it looks? Right, it's again 40 bases as well. The length of your sequence is 40 bases, but starting from position, let's say position 18, 19, all the way to the end of your sequence, end of your fragment, you have quality scores which are below 20, which is not acceptable. Okay, so if you want to continue analyzing using this data, you will need to do some trimming of the ends of your fragments. You may end up getting even shorter fragments because they are really poor quality. So you might end up getting just fragments of 20 bases, but it is better to use shorter fragments which you are confident of that no, it's accurate, than using the entire long 40 bases, no, the entire fragment which has half of it which are inaccurate, right? So with this, you can see that a lot of trimming is required. Almost half of your almost half of each fragment needs to be trimmed off because it's very, very poor quality. Okay, and right, the next one I always look at is your overrepresented sequence, your second last one. Okay, so this is how it looks if you have overrepresented sequence. Okay, this is the sequence. They show you what's the sequence, how many times it occurs in your data, and this is the percentage in total, right? Over your data, in your data, 0.5% of your data is actually this sequence. However, possible source, no hit. So if it's no hit, you don't have to worry. But if there is a hit, yeah, if there is a hit with Illumina primers or adapters, it will show you. Okay, so in this case, you will need to do some trimming to get rid of this sequences. I mean, you're not getting rid of the entire sequence. You're basically just trimming the sequences which has the primer. You're just trimming the primer portion of the sequence, right? So it's very important to do these three things when you get your data, when you do your QC, right? Three things that you always look at is basic statistics. This table, it will give you an idea of how many fragments were sequenced, what is your sequence length, percentage of GC, and also whether there's any sequence flagged as poor quality. But this is normally not very accurate. We always don't rely on this. We always go to the second one, which is your per base sequence quality, right? So this is very important to look at to give you an idea what kind of analysis pre-processing you need to do before you can start your analysis itself. You will need to trim, you will need to get rid of some portions of your sequence. And the third one, which is really important as well, is your overrepresented sequence, right? All those with no hits, you don't have to worry, as you must look through the entire table to see if you have any that matches Illumina primers or adapters, right? So that three things are the main important criteria you must look at. Basic stats, per base sequence quality, as well as your overrepresented sequence. If all three are good, okay, you don't have any overrepresented sequence, and your quality scores are, for instance, at least above 22, 25, you are good to go. You don't have to do any trimming at all, right? And your basic stats just gives you an idea, right? These two, per base sequence quality and overrepresented sequence, will tell you whether you need to do any trimming or pre-processing at all, right? So that's just to show you what we do normally once you get your data. What's the first step? I will continue sharing my screen. Okay, so once you've done your quality checking, and if you see that it requires some sort of trimming, or you need to eliminate some parts of your data, or you need to filter some of your reads which are poor quality, there are a lot of softwares that can help you do that. You don't have to manually look for the primer sequence in your data and then delete. You don't have to do that. It helps. A lot of softwares are there which can help you do this. Okay, and right, one of the most widely used, I would say, software is called Trimomatic. They also have others called CutAdapt, FastB, others which can help you do the same thing. And this is just to show you how a command from Trimomatic looks like. Okay, you don't have to memorize these commands. When you go, okay, let's say you want to start using a particular software, you need to go to their manual. The first thing I do is go to the manual, and they will tell you the default commands and all the different options or parameters which are available. So you basically need to know what you want to do, right? So this is the command, default command that's given. You need to know whether you have paired reads or single end reads. Okay, paired end, when you're already sending it out for sequencing, you will know whether you're going to be doing a paired end sequencing or a single end sequencing. Usually, most researchers do a paired end sequencing to be able to get a more in-depth coverage and a more accurate data, right? So the first is to just ensure that you know that whether it's paired sequencing or not. And then you will need to, for instance, let's say there is an overrepresented sequence. There is an Illumina primer, which is actually Illumina primer sequence which is actually observed in your data, you need to use this parameter called Illumina clip. Not parameter, it's an option. You have to use the option called Illumina clip, which will help you perform the following. Okay, so it will help you actually remove the adapter provided, and so if you just put the word Illumina clip and also mention the name of the adapter or primer, it will look through your entire sequence data you're giving to clip off or remove the sequence, right? And so basically this is how it looks like, just to give you an idea how a command looks like. Okay, and the last one, min length. So you're basically telling the software to, the minimum length of the reads that I want to keep is 36 spaces. If anything shorter than 36 spaces, get rid. Okay, so you can actually set your own minimum length for in your data before you can continue your analysis. Okay, so all this actually, determining which Illumina clip, meaning which adapter to be clipped off, how short your data, how short fragments you don't mind working with. All this depends on your QC, your initial QC. So once you get your data, you need to do your QC, and you'll have an idea of how your data looks like and how much you need to trim or what you need to trim. All that can be decided after you do a QC. Okay, so once you've done trimming, you can choose any of these softwares. Trimomatic is just one. You have CutAdapt, FastP, and a few others. Let's say you've done your pre-processing, you've trimmed off your primer sequence, you've filtered off poor quality reads. All that is done. The next step is again running it through FastQC to check whether your data now looks good, right? So you remember I just showed you how FastQC, how good Illumina data in FastQC looks like. So once you've done your pre-processing, you should run that again to see if it has improved, right? The quality, if you have done this trimming, your quality should have improved, and your overrepresented sequence will not have the primer sequence, showing up anymore. And if all that is good, you can then proceed to your next step of analysis, right? So analysis, it depends if you're going to be working with genomes or transcriptomes. You have different workflows, of course. So that depends on what kind of data, what kind of analysis you want to do. But this is the default analysis that you need to do before moving on to assemblies or annotations, right? The moment you get your data, you need to do a QC, you need to do pre-processing, and after pre-processing, do a QC again to be sure your data is good, and then you start your actual analysis. Okay, right, so that's all actually for today. Do you all have questions? Is there anything you want to know or want me to repeat, or are you able to follow? Right, you can actually unmute yourself, because if you don't write, if you don't talk, I won't know whether you can follow, right? Right, so since everyone is very quiet, I assume all of y'all understood. And so yeah, my next question, are there anyone who still hasn't been able to get their virtual box up and running? I know there was one student who wrote to me so far who hasn't got. Anyone else having trouble with your virtual box?
Miss, in the virtual box, I can't found this file because I follow the video, right? But something is missing, so I cannot.
You're the one who wrote to me, right? Just before the class? Yeah, okay. Then I'll write. I will follow up with you after this.
Okay.
Anyone else having trouble? So I assume, yeah.
Doctor, I also have problem, doctor, to download.
Who's this?
Sangita.
Okay, you're the one who wrote to me already as well, right?
Yeah, doctor. Yeah.
Okay.
Me too, Miss.
Who is this now?
Priscilla.
Priscilla. And your problem? What's your problem?
I downloaded it, but then I couldn't open it.
Okay, you guys write to me so that I'm able to fix it, because this Thursday we will be doing a practical on Unix. So it'll be good for y'all to have your virtual box up and running so that y'all can practice after that, right? So make sure to contact me. Right, Shazatol as well. Not able to download. Okay, so contact me, just message me, and then we'll try to fix it. If I can't, I will speak to Dr. Siva, and he'll also help you all fix it. Okay, and okay, I think that's all. Thank you. And the bioinformatics students, can you all just please stay back? The rest of your biotech students, you can. That's all for today. Thank you.
Thank you, Miss.
Right.
Thank you.
Right. We have. I just want to brief a bit about your project. The bioinformatics students, I just need you all to be around for a while. Okay. So for bioinfo, you have another project that you need to do. Okay. So one is the group project that is similar to the biotech students. For your, we need to have another project, and I was intending to have you all work on meta-transcriptome and metagenome, right, analysis. So we have 5 of y'all, and so one group can have 3 students, the other can have 2 students, and the one that's going to work with metagenome, I would like y'all to be in a group of 3, and the one working with metatranscriptome in just the 2 of y'all, right? So the first step is to do a literature review. Those who are not, biotech students, you can leave. I don't want y'all to get confused with your project one, because this is only for bioinfo students. Manoshini, I think you can leave as well. And Fazla, I don't know who else. Chung Lishan, I think you can also leave. All right. So you have a separate project, as I mentioned. The first step I would like y'all to do is do a literature review. So I want y'all to, I will send y'all a table, like a template of how your literature review is supposed to be. I want y'all to basically do some reading, see what kind of analysis, what kind of data they use for metatranscriptome or metagenome, what kind of workflow, right? What is the first step? What's the next? And for each workflow, right? For each step, what softwares are being used? Okay, so I will give you all a template of a table, and I want you all to work on I want you all to do a literature review first. So the first 2 to 3 weeks, maybe up to Deepavali, right? So you have a semester break the Deepavali week, I think you have a semester break. After that, on Monday, y'all can present your literature review of findings, and then we'll move on from there. What I basically intend to do is give y'all a small data set, and y'all can do some analysis with it based on your literature review. So you will need to come up with your own, not your own workflow, basically look at journal articles, see what is the common softwares that are used, and we will pick one workflow with simple softwares that don't use too much RAM. We'll pick that, and you can do some analysis, right? So that's the whole idea. And I hope y'all don't have a problem with your virtual box. Any one of y'all?
Only me, Miss.
Okay, right. The rest of y'all okay? Then so So that's it. Basically, I'll give you all a table and I need you all to start working. Who are the 3 and who are the 2? I need 2 groups. So
Hovinia, Savitri, and Priscilla, 3 of them in a group, and then Shad, Lo Bingfeng, 2 of them are in another group.
okay, right. So the one with 3, you all need to work on metagenome, right? And metagenome, there are 2 kinds. You have shotgun metagenomics and you have 16S metagenomics. So you all will need to basically do some reading and look at the usual workflow and what kind of softwares are used. And I also need you all to get the link for these softwares, whether it's a GitHub page or if it's a web server, then the web server's link, right? So you need to do both 16S as well as shotgun. So the 3 of you all have to start working on that, do the reading. And Prashad and Bingfeng, you guys need to work on meta transcriptome. So I need you all to go and do some reading on what is the usual workflow, what are the softwares used. Again, I need the links for web servers if there are web servers, and links for GitHub page, right? So,
Okay.
so that's it for today. We'll send a table and y'all can start working on it. And I guess I will need y'all, once you've completed your table, I will tell y'all when, maybe in 3 weeks time, I need y'all to do a short presentation on what you found, okay? And it will just be a small group, just us. And once you've done your presentation, then I will give you all a small data set and you all can start working on that. And I also have a few tutors that will help with this. So we have my PhD students, Mahin and Sumitra, who will be guiding through the meta transcriptome students. And I have Jason and Jared, who will be helping with the metagenome group, right? So they will also be part of our presentations and discussions, and you'll see how it goes. They will be helping as well. Okay, so we'll see you all this Thursday for the practical, and the few of y'all who have a problem, I will contact y'all separately. Right? Thank you.
Okay, doctor. Thank you.
Okay, bye.
Thank you, Doctor.
Okay, bye. Bye.
Bye, Miss.
Bye.